Yeah, sorry, I don't know where. I don't know if there's like the USB thing for that. You could have it like this match. This like extends. So we'll just set it here as you're speaking. Okay. And then control from the keyboard. There's also a microphone if you like. Is it on? Sorry. Okay. Yeah. Right, this should be it. I'll be in the back over there in case you need it.
Okay.
No, people enjoy it.
Yeah.
What is your PhD thesis on?
My PhD thesis on soft materials for neural regeneration.
I see. So your degree is in chemistry?
In actually, because it's in Germany, so it's natural sciences.
I see.
But the institute was polymer science,
I see.
polymer research.
Science.
Yeah.
Okay.
Trying to figure that out. I've applied for a few jobs. Yeah. Yeah. I'm working on it. I applied to Wolf Speed, actually. So I can work on the power semiconductor. I would also apply to. I forgot their name, but it was energy storage,
Ah, okay. Sure. Yeah. I don't know. Can you hear me in the back or? Can you hear me in the back? I don't think this is working. Okay. Ah, okay. Sure, no worries.
Okay, I guess I'm not gonna wait. So today is our pleasure to have Dr. Aleeza Farrukh giving a seminar today, and she's a postdoc at UC Irvine. She got a PhD from Max Planck Institute in Germany in polymer science.
Yeah.
And so she's going to talk on biomaterials to bioelectronic devices, precise control over cell fate. So let's welcome Aleeza.
Thank you for the kind introduction and for this opportunity today. So what I'm going to discuss today is basically like how can we use different biomaterials and bioelectronic interfaces to control the fate of cells. So I will start with a brief introduction about myself. I hold a diverse and research and education background with MPhil in organic chemistry and PhD emphasized on polymer chemistry. So during my research training, I work on different areas, started with organic synthesis, then polymer synthesis, nanotechnology, biomaterial engineering, and currently I'm working on organic electronics. So going back to the cellular microenvironment, I will start with a very brief introduction what cellular microenvironment is, so that we have a context of what I'm going to share today. So starting with that, basically cells in the body, they are in a very complex environment. They have other things surrounding them. So they are not like singly like existing in a body, they have other things surrounding them. And this environment which is surrounding the cell is called a cellular microenvironment. So what is there in the cellular microenvironment? Of course, in the cellular microenvironment, there are other cells, and the major component of cellular microenvironment is the matrix which surrounds the cells. So on one hand, this matrix provide the support to the cell, like physical support to the cell. On the other hand, it does have chemical interactions with the cells. So with this, we see like from cells, from other cells, there are also a lot of growth factors, soluble factors released from the neighboring cells, which are part of cellular microenvironment. And then there are physical forces. So the extracellular matrix, so ECM is actually extracellular matrix which surround the cells. So what happened that extracellular matrix itself stretch on the cells, and the other neighboring cell also have some tension on the cells. So just to summarize everything, in a cellular microenvironment, there are neighboring cells, there are extracellular matrix around the cell. And these extracellular matrix and neighboring cell give chemical cues as well as mechanical cues. So what happened due to this cues? So what does they do? So what happened is like when the cell receive this cue, depending on what is the message from the other, from the environment and from the other cell, it can either decide to differentiate, it can either decide to die, like have apoptosis, it can decide to have biosynthesis, it can migrate. So that's why cellular microenvironment is extremely important to control the fate of cells. But then comes the part like, why are we interested in, like, why do we care about fate of the cells? So we care for fate of the cells because, of course, like in the body, starting from a single cell, they divide into different cells. And depending on their extracellular microenvironment, they decided to differentiate either as muscle cells and make muscles, as neuron cells and make brain, or as bone cells and make bones. And even during the growth, like now, every day, if we have some injury, if there is any defect, and all these things, they are actually controlled by cellular microenvironment. So then comes the question is like, how is it relevant to material chemistry or material sciences in general? So basically, as I said, that the cellular microenvironment have two main components. There are these biomechanical signals and biochemical signals. So from ECM, cell is receiving this biochemical signals. So in extracellular matrix, actually extracellular matrix is a mixture of different proteins. There are structural proteins, and then there are fibronectin and laminin, which are more like an adhesive proteins. So what happens, like from material science perspective, there are chemical cues functionalized on this matrix, which give message to the cells, and also there are mechanical cues coming from the matrix itself. Similarly, we have messages coming from the cells themselves, the neighboring cells. So there are growth factors coming from the neighboring cells and mechanical forces coming from the neighboring cells. But the interesting part about this is like everything is very dynamic. It's not a static environment. It's not going to have the same mechanical and chemical cues. And that is the part of extracellular matrix which make the difference, so which helps in making the different response from the cells. So when we try to engineer this material in our, like if we want to have an artificial material, we want to create a material for tissue regeneration or engineering, there are different cues that we need to actually recreate. So what do we really need? So if we want to mimic the cellular microenvironment, we need a chemical functionality. So we need a protein which actually are going to support the growth of cells, or we need growth factors which are going to dictate what's going to happen with the cell. Then we need a mechanical properties, like what is the stiffness of the material, what is the topography of the material, and finally we will have some structural feature, how the material is arranged. So if we want to recreate the cellular microenvironment artificially, we have different factors to recreate. And it's not stop here. So then comes the interesting part that we need to do is like dynamics. So in addition to having like specific functionalization, in addition to having specific stiffness and topography, we need to have a dynamic control over all these factors. So then we can choose when we want the cell to receive certain cues, and we can control when we don't want the cells to receive certain cues. For example, if we want the cell to grow, so we will release the growth factors and that can stimulate a cell to grow. On the other hand, if we want the cells to migrate, we can show it different chemical cues on our surface so that it can migrate. And this is a part in artificially recreating the material for cellular microenvironment, which is still very challenging because there are a lot of factors to consider. So I think with this brief introduction about cellular microenvironment and cell fate itself, I can go to the projects that I'm going to share today. So in first example, I will share that how we can electrically control the release of growth factors from one cell on top, that they are going to affect the differentiation on the other cells on the bottom. And in the other example, or in the other part of my talk, I'm going to show that how we can selectively show cells certain chemical signals, so they are going to migrate or differentiate in response to those chemical signals. So I will start with bioelectronic control over angiogenic differentiation. So basically in cell, as I said, there is one cell which we are calling source cell, and then we have a recipient cell. So what happened that source cells, it's going to release some chemicals that we are calling like growth factor is going to release, but mostly it doesn't release it like directly. Sometimes it does, but mostly it packages it into these vesicles. So these are like micro vesicles actually like less than 1 micrometer. So they are called extracellular vesicles. They are between 50 nanometer to 1000, 50 to 1000 nanometer. So what the cell does, instead of releasing it directly into the environment, it packages it into these vesicles. So what happens that these vesicles can pick up by the recipient cell, and because they have receptor on surface, they can pick up by the recipient cell more specifically, and then they like release whatever growth factors they have, and they can control the response of recipient cells. So either it can trigger it to migrate, it can trigger it to differentiate, it can trigger it to like undergo apoptosis. So it can control different factors. So this makes extracellular vesicles very interesting in terms of therapeutic applications, because we can package whatever growth factor we want in the extracellular vesicles, and then we can put it to the source where we want them to like go, and they can specifically bind to our recipient cell, and they can trigger the process that we want to trigger. But when it comes to using extracellular vesicle as a delivery vehicle to transfer some cargo, then we have some problems there. First, because these extracellular vesicles, they are released by the cell. So which means that we don't have any control, like what is their size, we don't have control what is the composition, what is being packaged in the cell. We don't have any control how many of them are released. And then we have an issue like when they are released, how the recipient cell is going to respond to these growth factors. So from artificially controlling the extracellular vesicles, there are different methodologies. So there are different technologies which are trying to somehow control how is the composition and how is the concentration of vesicles. So there are strategies which are either using genetic engineering to trigger production of extracellular vesicles, or some external stimulation, for example, electrical stimulation or acoustic stimulation. So what does those stimulation does that they destabilize the cell, and when they destabilize the cell, so cell try to counteract it and release a lot of these vesicles outside. And then there are some chemical cues, for example, using some hormones or using some like pH. So at low pH, because cell have this proton pump, so when you reduce the pH of the environment, it's also going to trigger the cells to release these vesicles. So in principle, there are different methods to do it. But what we can see is in all of these methods, there is no control over the composition and sizes. So they do have some control in terms of how many exosomes are being produced, but they don't have any control when, what is like packaged inside, and how is the size of the vesicles. So to have a controlled production of extracellular vesicles, we get some inspiration from another species and actually another class of animals, and that is cephalopods. So cephalopods are basically really they have very impressive skin architecture. They have a lot of different types of cells in their skin. And what does they do? It control a lot of things like their visibility, their camouflaging, and so on. But the part of the skin cell that we are interested in are leucophores. So leucophores are type of this cells in cephalopod, which actually control the like dispersion of light. But the most important part that we are interested in leucophore is actually this vesicle. So leucophore produce a lot of these small vesicles, and in a very controlled size and in a very controlled dimension. How does it do that? It do that because it have like a protein, a special protein, which is called as reflectin. So these proteins, they have a lot of aromatic amino groups and a lot of charge. So this protein can reconfigure. It can either form nanoparticles, it can either form sheets, and it can either make like different structures based on the environmental cues. So basically, how leucophore control the production of a very well-defined vesicle is by using this reflectin protein. So we get our inspiration from this leucophore architecture. And in our group, it was established that we can actually engineer human cells to produce the reflectin protein. So when the human cells produce the reflectin protein, it was also packaging it in form of extracellular vesicles and particles within the cells. So this is part one. But the interesting part is when we apply any stimulus, because it's a charged protein. So if you change the ionic concentration, if you change the pH of the system, then we can trigger the production more, and we can trigger it to release these vesicles. So this is how it does. So here you can see the cell. You can see these reflecting particles which are within the cell. These are the small vesicles containing reflectin within the cell. And when we apply a stimuli, we can start to see these vesicles being released out of the cells. So to prepare our cells where we have control over the production of exosomes or extracellular vesicle internally, we decided to use this system. But then the interesting part is how do we control it? Like where is the controlling part? Yes, we can produce extracellular vesicle in human cells, but how are we going to control them? So as I said, this reflectin protein, it is responsive, it's a pH responsive protein. So the control that we wanted to have bioelectronically is to use a proton conducting membrane. So by using a proton conducting membrane, we can change the local flux of the proton on the surface of the membrane, and we can actually trigger the production of vesicles which are reflecting based. So just to summarize it, so this is the devices that we have with these are the bioelectronic device. So what do we have here is we have a membrane which is proton conducting coming from our proton conducting devices, and we have cells which we have genetically engineered to produce reflectin. So when we apply a bias, we have a proton flux across the membrane. And what this proton flux does, it's going to change the surface pH, and then it will trigger the cells to release the vesicles, which are like reflectin containing vesicles. Okay. So now we have our setup. So how does the setup work? How do we really control it, and what can we do with this? So with this part, what we have is that we first of all genetically engineer our human cells, and you can see the reflectin in the mammalian cells. So these are human kidney embryonic cells, and you can see that the reflectin, the purple ones are within the cell, and in TM images we can also see, because this reflectin protein have very high charge density, so you can see them also in the images. Then comes the part where we prepare our devices. These are typical proton conducting devices with a semicircle and a spur. And when we apply a bias, and we wanted to see if we can really changing the surface of the pH by applying a bias. So this is when we don't have a bias in the start. So we don't see anything or like any charge on the surface, and the surface is more or less neutral. It's around 7.2 pH. But when we apply a bias of 500 millivolt, what we are seeing that there is a charge in proton accumulation on the surface of the protein or of the membrane. And this charge accumulation result in drop in the pH to 4.2. So with this, we have our device, bioelectronic device, which is proton conducting, and we have a cells on top, which is pH responsive. So what can we do with this? So what happened is, like when we apply a bias of different voltages, so between 100 to 800 millivolt. So now I just want to emphasize a point, the electroporation, that the technology that is available in the literature, they are using 50 to 200 volts. We are using 100 to 800 millivolts. So when we are applying this bias, we can see that there is increase in the concentration of the vesicles around 500 nanometer. How do we know that? First of all, these are the immunofluorescence image of the cells. You can see this pink protein within the cell. Then we can actually collect the extracellular vesicles from the media of the cell, and we can use nanoparticle tracking. In real time, we can see how many particles are there, what is their size, what is their concentration, and we can even do the immunofluorescence on those particles and see what is packaged in the particles. So within the particles, so all the particles, they are marked within RNA protein, which is green. And then you can see a lot of purple dots, which is actually reflecting protein packaged in the extracellular vesicle. So with these techniques, we can see that when we apply a bias between 100 to 800 millivolt, we have a peak production of reflectin containing extracellular vesicles at 500 millivolt. we can also see that we have a peak packaging of the reflecting protein at 500 milliwatt, which means that when we apply a bias of 500 milliwatt, the reflectin is actually getting packaged within the vesicles, and we have more than 80% packaging of the protein in our vesicles. And finally, we also have some control over the size of the vesicles, and this size is increasing with increase in the bias, because of course, the more you destabilize the cell membrane just by having the pH outside the cell, then the more it's trying to get rid and destabilize its cell membrane by making these particles, which are basically reflecting containing. So then comes the part that it's only relevant if we can have this control over multiple cycles. If we can only produce vesicle once with electrical actuation, it is interesting, but it's not very applicable. So what we wanted to see is, can we have this control over multiple cycles? So what we did, like we have our own cycles, all of these cycles are 2 hours. So we have 2 hours of on cycle, 2 hours of off cycle, and they are sequentially, we were trying to do it. And then we will see like what happened. So what we observe is, like when we have an on cycle, we have a significant increase in the production of extracellular vesicle. When we have off cycle, sequential off cycle, the production is decreased. And again, we can do it at least over 3 multiple on and off cycles. So what do we have here? So we have a system, a bioelectronic device based on the pH responsive protein, where we can control the number of cells. So in the number of extracellular vesicles, we can control the concentration of extracellular vesicle, we can control the composition of extracellular vesicles. And we can do that at very low voltage, and we are not killing our cells. So our cells were maintaining their viability. All of these electrical bias, they were cell-friendly. And we can do it over multiple cycles. Okay, so this is good. We have a system where we can have some control over the vesicles. But what we are going to do with this system, how can we use it for anything relevant to tissue engineering or anything relevant where we can like really apply our bioelectronic devices? So there we go, that we need to, so reflectin protein itself is just a pH responsive structure protein. It does not have any chemical cues. It's not going to trigger cell to do something. So to have the certain response from the recipient cell, what we are trying to do is that we are trying to couple it to a growth factor or a signaling protein. We can do any signaling protein coupled to reflectin, because we are going to genetically engineer our cells to produce this protein. So in our case, we selected VEGF. So what is VEGF and what is angiogenic differentiation? So first, I will give a brief introduction about angiogenic differentiation. So angiogenesis itself is blood vessel regeneration. So within the body, when you have any injury, even in journal, there is some like the blood vessel, they keep on forming, all blood vessel, they disintegrated. And this process is called as angiogenesis, like blood vessel formation, new blood vessel formation from existing vessels. How does the cell do it? So cell release this vascular endothelial growth factor. It's a growth factor which control the blood vessel formation within the body, which is we call VEGF, basically. So in a lot of tissue regeneration therapies, when you get any implant, we always need a blood vessel and circulation around the implant. Otherwise, the implant is going to get like, it's not going to get any oxygen, and it's going to disintegrate and die at a certain point. So that's why VEGF is extremely relevant in terms of tissue engineering therapies. But there is a problem with VEGF. The problem is the dose. If we don't have enough VEGF in our system, the blood vessels cannot regenerate and cells are not going to get enough oxygen and they are going to die. If we have too much VEGF, it's going to over-trigger the vessels and the cells are going to become like tumor-like cells. And this is what happened during, like if there is a tumor within the body, mostly there is very high like intensity of blood vessel around that, because they need a lot of oxygen to grow. If we want to use VEGF to make like in tissue engineering, we should have a control over their dose. And this is what we are trying to attempt with our bioelectronic device, because we have a very good control over the composition. So what we are trying to do is like, okay, so if we can make our cells to produce VEGF, And we can externally control how much of VEGF it's being produced. We can control the blood vessel regeneration. So step one, we genetically engineer our cells to produce VEGF. Now you can see there are two different colors. We have reflectin, which is red. We have VEGF, which is reflectin, which is pink, and VEGF, which is red. And these are produced in tandem with each other. So if reflectin is being produced, we are also producing VEGF because they are fused. And they are the vector for production is like fused with each other. So if they are producing one, they are going to produce the other automatically. Same thing, we can collect the vesicles, we can do the nanosite, we can check under immunofluorescence what is packaged within the vesicles. And here we can see that when we are increasing the voltage, we are increasing the number of vesicles, which is not surprising. This is something we would expect. We can increase the number of size of particles, but here it gets very interesting. So when we have a 500 millivolt actuation bias, we can produce the VEGF and RFA1. But if we don't have RFA1, if we only have the cells which are just genetically producing VEGF, they are having the peak at 100 milliwatt. So basically with the applied bias, if we are using 500 milliwatt, if we are using 100 milliwatt, we are really controlling the composition, what is being packaged within the vesicles in the end. So, okay. So now we have a system where we have a control over the composition and size and concentration of vesicle. How are we going to use it? So to use it, what we are trying to see is we are trying to replicate this cell-cell interaction, which I showed that there is the cell-cell interaction. So during cell-cell interaction, these growth factors are produced by one cell and the other cell respond to these growth factors. To replicate that system, we have a dual-tier device. On the top of the device, we have the cells which are going to produce the growth factors. On the bottom of the device, we have the cells which are going to respond to these growth factors, which in this case is endothelial cells, basically, human embryonic endothelial cells in the end. So what happened that if we don't have any applied bias, they are just sitting on top of each other, there is a semi-permeable membrane which does only allow the vesicles to go through. It does not allow the cells to go through, basically. So the vesicles, like you can see that there is one vesicle, it's going down, and there is the endothelial cells. So we can see they are making the cell-to-cell contact. There is nothing special at the moment. There is no differentiation. But when we apply a bias in situ, in the presence of both cells are there, we apply a bias. So now the vesicles which are released by the top cells, they are going to trigger the differentiation of the cells on the bottom. So they are these endothelial cells, they are going to have angiogenic differentiation, and they're trying to make blood vessels in the system. And we can see that we have very good control. So when we have the vesicles which are producing both RFA1 and VEGF, we have very good control over the angiogenic differentiation. So I hope that till now I can show you that how by controlling the cell-to-cell communication, chemical communication, we can electrically control the differentiation of the recipient cell. And we have got the grant on this work from NIH to take this device and make it like for in vivo applications for blood vessel regeneration in mice. Okay. So this was the first part of my talk. Now I will go to the second part of the talk where we are trying to see how can we control the chemical cues from extracellular metrics to control the different direction of a cell, basically. So what we are really interested in are neural cells. So we are trying to replicate neural regeneration. Why are we interested in neural regeneration? So when there is any injury happen to brain or a spinal cord, generally it's known that it's almost irreversible. You cannot cure it. The main reason is that in brain and spinal cord, there are not a lot of stem cells. So these are the part of the body which contains very low stem cells, which mean if there is something happened, if there is some injury, some cell die, there are no other cells which can actually regenerate and recreate like they can sort of cure the injury. So in neural regeneration therapies, what people are sort of projecting is that if we can inject the cells, we can take the stem cells from some other part of the body or from some animal or some other source, and we can inject at the site of the injury. And in principle, it should work. They are cells, now they should be fine. But there is one important part of neural regeneration which is being ignored, that we need a circularity. Like in neurons, one neuron is going to attach to another neuron. So basically the terminology is neural rewiring. And it's very similar to any rewiring, that one circuit needs to be connected to the right circuit. Otherwise will be no impulse going from one cell to another cell. So this is a part which is still very difficult to control for neural regeneration therapies. The part is direction of the neurite. So how are you going to redirect the neurite? So when we look into the material part, how can, from material science point of view, how can we control the direction of neurite? So in literature, there are different strategies to do that. Very straightforward strategies to have some topographical features. So if you have a certain topography on the surface, or if you use electrospun fiber with certain orientation, you can in principle trigger the cells to grow in that direction. Why? Because cells need a support to grow, and they are going to grow in the direction where, however, the support is sort of predicting them to. Then there are other way of doing it, which is basically using adhesive protein. So as I mentioned, in cells, in extracellular matrix, there are these adhesive protein, which actually allow the cells to attach and to grow. So the other strategy is to use this adhesive protein, like either doing microcontact printing or 3D lithography to have them oriented in a certain way. And we can have some control over the direction of neuron. But what is missing here is the dynamic part. We cannot, like, these are static systems. There is nothing dynamic. So if we, for example, inject cells in the brain and we put them on a surface which is like already aligned, they are only going to grow in that direction. But how do we know? Like, we want to reconnect it. We don't know what is the direction of the neuron where we are trying to reconnect it. So the part which is missing is spatial orientation with temporal control, that we want to see which neuron is going to connect to which axon and how we are going to do that. So during my PhD in Professor Del Campo's lab in Max Planck, we have a strategy of controlling the activity of peptides by light. Why light? Light is a very interesting stimuli because you don't need to inject light. You can just shine light through the skin and you can control different part or different behavior within the body without any invasion in the body. So how does this system work? Like, what is the light responsive biomaterial? So in this system, first of all, we start with the selection of a biomolecule. For example, if we want to grow neuron, we are interested in laminin protein because this is a part of protein which is present in the extracellular matrix of neurons. So we can select a certain sequence instead of using the full protein. We can actually use the full protein, but it's better to select the part which you are interested in. In our case, that was a part which is like ICVA 5 amino acids. So first of all, we select what is our biomolecule of interest. Then we add a photo-removable group. So we put we take a group which can be we take a protecting group which can be unprotected with light. And we put this group on the active side. So what happened, like when this group is there, then this protecting group is photoprotecting group is there. The cells, they cannot see the protein, they cannot recognize the protein, as long as this photoresponsive group is there. And this photoresponsive group we can uncage when we want. So once we have our peptide, we can select to put it on a surface. Mostly if we are working with neurons, we are going to use some hydrogels and we can functionalize it on top of hydrogels. And then we can shine light. So the cells, the part where we are going to shine light, these photo caging groups, they are going to get removed. And now the cell can see this protein and they are going to align or going to respond in those areas, basically. So, okay. So this is the strategy that I will show you today, that how can we control direction of a neuron by using a photoresponsive ICVAF peptide. So first of all is the selection of peptide, which I have sort of already covered, that we select this ICVAF peptide. It's a 5-amino acid peptide coming from the laminin protein. And this is a peptide which actually control the control the attachment and growth of neurons, basically. So once we have the ICVAF peptide, next thing is to put the photocaging group. So in the case of ICVAF, the active site is lysine amino acid. So when cell try to see where is the protein, it's try to recognize it with lysine amino acid. So what we decided to do is to try to put the photocage at lysine amino acid. So this is how we synthesize it. So in our case, we selected this nitrobiphenyl based photocaging group. So what is important about this photocaging group is like we can either uncage it by using 1 photon laser at 405 nanometer, which is cell-friendly wavelength, or we can even do 2 photon actuation by using 800 nanometer, which is like really cell-friendly, and it can penetrate very deep inside the tissue as well. So first of all, I synthesize this caged lysine with this photocaging group. Then we do the peptide synthesis. We can synthesize this peptide with this lysine amino acid. And then comes the part where we can, like part 3, that we can functionalize into any surfaces. So here we run into a problem that we are trying to work with neurons, which means that we need a very, very, very soft surface. Brain itself has a very low stiffness, so it has something between 0.3 and 1 kilopascal. So what we decided to use is like, yeah, we can use polyacrylamides, we can tune the stiffness, they're very cheap, they're very easy to do. But there is a problem with polyacrylamide gels, this amide group. Amide group is not very reactive group, which means that you cannot covalently functionalize anything on this gels. We can do some coating, but it's not very helpful. So what we tried to do was like we wanted to couple this ICVAF peptide covalently to polyacrylamide hydrogens. So to do that, I designed 2 different new acrylates based on methyl sulfone chemistry. So this is the part, this is like methyl sulfone. So what happened that I designed 2 new acrylates where we can have them copolymerized with a polyacrylamide hydrogens, and then we can functionalize our peptide at room temperature in PBS without any issue covalently. So in first step, I try to copolymerize it with polyacrylamide, and what we can see, it does not affect its properties. So the polyacrylamide still does not have very high polydispersity. The swelling was fine, elastic modulus was fine, and elastic modulus was the part which we are more interested in. So now we can see that these methyl sulfone based acrylate, and we were only using 2% of it. We don't need like a lot. We are just trying to do the surface functionalization. So by using 2% of methyl sulfone acrylate copolymerized with polyacrylamide, we can actually have a like polymer where we can covalently functionalize biomolecules on the surface. So once we had the polymer, we did the next step, which was making the thin films. First of all, we made the thin films, and then we try to use like fluorophore just to see like if we can really do the coupling. So when there is no methyl sulfone here, we can see that nothing is getting bind onto the polyacrylamide. But when we have methyl sulfone containing co-monomers with our polyacrylamide gel, we can have quantitative binding of these fluorophore or the chromophore on the surface of the gel by using this methyl sulfone thiol chemistry. So now we have our peptide, we have the cage version, we have the hydrogen. So what we are going to see before putting cell on top, we want to make sure that we can do the uncaging. So what we did is like we can do the uncaging in solution, and we can do the uncaging using 4 or 5 nanometer laser. So this is what happened, like the longer you do shine it with light, the more photo cages going to get removed. And we can have 86% of removal of this caging group in 15 minutes. So now we come to the final part where we are going to interface this surface with the cells. So we are going to use neural stem cells, and we will see that when we have neural stem cell and if they can see EQUAF, they are going to grow. So just to give a recap that how does the neuron look like? So neuron have an axon, neuron have a neurite, which is like the longer neurite is called axon, the short small neurites are called dendrites. So if the cell can see the EQUAF peptide, it's going to grow and make these neurites and dendrites. So just before going towards the actual part, I just wanted to show you the effect of stiffness. So this is how neuron look like it on glass, and this is how neuron look like when it have the right stiffness. So stiffness of your system makes a lot of difference. Okay, so now we have our EQUAF peptides like on the surface of polyacrylamide hydrogels, and it is caged. And we put neurons on top. What will happen? Neurons are not going to grow because they cannot see the peptide. So you can see that there is just this blob of nucleus. There is nothing else because the cell cannot see the peptide. So in next part, we wanted to do in situ activation, which means that we are going to uncage the peptide in the presence of cells. And then we are trying to see if we can control the growth and direction of that neurite. So what we did here is basically, so this is one cell. So this is how neuron look like. These are before growth, they are just small circles. And I shine the light in front of this. This is 100 micrometer irradiated area, and this only takes 10 seconds using a microscope. So what happened then, the light is shine, neurite starts to grow. Generally, it takes 24 hours to grow, and it's growing only in the area where we shine the light. And when we did the immunostaining, we can see that this part is green, which means the neurite which we grow is actually axon. So with this system, we can control the growth of axon in situ using our biomaterial. But we cannot only just control the direction of the axon, we can redirect them. So this neuron is trying to grow in the other direction. So if we shine light, it will start growing in the direction of the irradiation. We can select, if the neuron have multiple neurites, we can select which one of them is going to become axon. Then we can also control the migration. So if the stripe that we are creating is a little bit big, it's if it's very wide and it can have the whole cell going through, we can actually control the cell migration. And the last part, which is the interesting part, which is the part we started with, was connection. So we can actually connect two neurites and two neurons by using our system in situ. So in the last section, this is something which was done on 2D surfaces, on 2D polyacrylamide hydrogels. It would be interesting to have it something similar in 3D. And just to have a proof of principle, I will show you how we can do the same experiments in 3D or similar experiments in 3D. But again, step one, we need a hydrogel where we can control the functionalization on the surface, where we can control the distribution of cells. So to do that, we see in literature, and we have this PEG hydrogels, maybe you have heard about these. These are really famous for making 3D functionalized surfaces. But there is when we are trying to make hydrogels where you have cells inside, it means that you need to do the cross-linking while you have cells. That means that the cross-linking chemistry should be cell-friendly. They should not end up in killing the cells. So in that case, we have two types of chemistry. There is thiol-maleimide and thiol-vinyl sulfone chemistries, but there are issues with both of them. So in thiol-maleimide, when you try to put the cells in the hydrogel and you are trying to polymerize it, it polymerizes within less than a minute, which means it does not let you have that you can mix your cells properly and then you let it polymerize. On the case of thiol-vinyl sulfone, what happened is it polymerizes really slowly. So you had your hydrogel, you mix the cells on top, and then it just takes 45 minutes to polymerize, which means that cells are going to settle down. And this is something you can see in the 3D stacks. In the first case, cells are just stuck on the top and there is nothing in the hydrogen. In the thiol-vinyl sulfone, what is happening, the cross linkage is so slow that everything is actually settled down. So then we try to use our 3D thiol methyl sulfone chemistry here, not only for binding the molecule, but actually as a cross-linking chemistry. So this thiol vinyl sulfone chemistry, in this case, the gelation happens between 3 to 5 minutes, which means that when you are trying to mix everything, you have enough time to mix everything. And then this gel starts to like gelate, and cells are really distributed within the system. So with this system, what we see that your surface functionalization and your chemistry that you are trying to functionalize your surface is extremely important. And if we can control that, we have a lot of control, not over just on the stiffness, we have control over binding, we have control over degree of polymerization. So just by choosing the right chemistry, we can have multiple control over our artificial material that we are trying to create. So just to show the last part, in this case, we try to use a different caging peptide because we wanted to see how everything looks in 3D. So here we are using RGD peptide, which is also a extracellular matrix containing peptide. So what does this peptide does is like it controls the migration of fibroblast, basically. So when we have the 3D gel with this caging group, the cells cannot grow. You can see that cells are dead. So the outer part is like these, every red spot is a cell. So on the outer side, there is some cells which are still alive, but inside the gel, the cells are dead. This is also tells you that if the cell does not get enough oxygen and enough supplies, they are going to die anyway. So when we uncaged it, when we have everything uncaged, there is no caging group. You can see the cells, they are migrating. So this small thing is a cell which is migrating out of these clusters. So when cell can see the protein, it can actually go everywhere and have the migration. So what happens when we have a caging group and we can selectively uncage it? So in this case, the cells, they are in a hydrogel, which is with this photocage RGD peptide, and their cells are not growing. You can see that this is like everything is dead. We have some cells which are there. But if we shine the light using a 2-photon laser at 740 nanometer over these cells, so this light is very cell-friendly, we can shine it over the cells, we can see the cell can now recognize this area and they are trying to migrate and they are trying to keep the direction of migration to the area where we shine the light. So with this system, by using photoresponsive peptides, we can selectively control the behavior of cells by not only like spatially, but temporally. We can see when, where, how we want the cell to respond. So with this one, we have spatio-temporal control over cellular migration and response based on photoresponsive peptide. So just to summarize my talk, in first section, I showed you how by controlling the growth factor using electrical actuation, we can control the differentiation of a recipient cell. And in the other case, I showed you how by controlling the signaling from the extracellular matrix, by mimicking the signaling from extracellular matrix, we have a control over the migration and direction of the cells. So with this research and educational background, in my independent research group, I want to control and prepare electroactive soft polymers. What does that mean? So my research vision is actually to start with the chemistry, that I can create different structure and different chemically defined chemistry and design of heteroaromatic monomers. And I can introduce different functional group like here, this R in this group, and I can control what is going to happen. So what I'm trying to do is basically that by controlling the chemistry of my monomer, I'm going to create different electrically responsive interfaces where I can either have a hydrogel, I can either have a shape memory polymer, or I can either have a pyroelectric polymer, which converts heat energy into electrical energy. So just by designing and synthesizing and controlling the chemistry of the monomer, I'm going to create different interfaces based on this polymer, and they can be used for a wide range of applications, for example, as a neural interface in soft robotics or as energy harvesting interface. So with this, I will like to, first of all, acknowledge my advisors at UCI, Professor Alon Grudeski, my advisor at Max Planck, Professor Anshadel Kempo, and my co-advisor, Professor Benedict Berninger. With him, I did all the neural related work. And I would like to acknowledge all of my current and previous colleagues for their cooperation during all this time, and all the funding agencies that supported this work. And of course, to all of you for your attention and for being here today to listen to.
Any questions?
Thank you for the interesting talk. It was around slide 34 or so. I think you showed neurons on substrates of different stiffness. And you commented that they were different. Could you tell us a little bit more about how they're different?
Sure.
I just didn't have long enough to see it well.
Yeah.
Yeah.
So when it's the right stiffness, you can see that you have very defined neurites, and then you have a long axon, especially this branching. So this branching is really important because this branching helps the cell, like the small red branches, it helps the cells to connect to another cells. So we call it like neurites, small neurites. But when we don't have right stiffness, especially on glass, there are almost no branches. And there are especially no branches on the axon. So even you have an axon, but it's not going to connect to another neurite because there's nothing to connect. So that's why stiffness on the other cases, when you have 20 kilopascal, which is still like way above the stiffness of the brain, but still it's not like glass, which is in megapascals. So we still see something. In all these cases, the surface is functionalized with the protein. So it's not that there is no protein. So everywhere the surface is functionalized. When we have very, very, very soft hydrogel, then what happens that neuron does not get enough like stiffness from the surface, and it's still having issues with their growth. So this is how it happens. Like if you have and for every body tissue, actually the stiffness is different. So that's why tunable stiffness in biomaterial is really important. Otherwise, we cannot use the same stiffness for everything. So this is how it is.
Okay, are there any other questions? Can you go to the slide where you show the impedance? I believe it's page 28.
Where I
Page
show
28, where you show the impedance analysis. It's 28. Is it 28? I believe it's 28. Oh, yeah, that's right.
Yeah.
18, sorry.
Yeah.
What kind of information you can get from the impedance measurement?
So here, basically, we are just getting the conductivity. So I can show you maybe the previous slide. So these devices that we have, they are proton conducting, which means that they are really dependent on the humidity of the environment, because of course there needs to be a proton in the environment. So what we are trying to do is we are trying to replicate is, and the conductivities of this protein is really humidity dependent. So when we increase the humidity, we have proton conductivity. So what we are getting this semicircle and a spur. So this semicircle actually represent the RC side, the circuit, where you have resistance and capacitance in parallel, and you get this bulk circuit from the membrane itself. And then this spur actually represent the electrode and the membrane membrane and electrode interface, because there what happens, like when we go to low frequencies, the charge starts to get accumulated on this electrode and membrane interface. And this is very typical for all proton conducting devices. It doesn't matter if it's a protein, if it's a peptide, if it's a polymer, all proton conducting device shows this very typical semicircle and a spur, because mostly the circuit is equivalent circuit is actually based on the, it's just a typical RC circuit in the end.
So why is this circle larger for the deuterium on the lower right hand corner?
So here what happened is like we were trying to see the kinetics. So basically what happened is like how fast a proton can move. So deuterium is basically more, it's slightly higher in terms of molecular weight. So it has low kinetics, like, which means that you will have high resistance. So like proton is going to transfer faster, and deuterium is actually 40% low in terms of how fast it can hop from one of this like device to another like part. So basically, so it's in the end, it related to the how fast proton versus deuterium can hop from one of the charge part to another charge part to another charge part. Because deuterium is high in the molecular weight, it have low kinetics of transfer, which means that you will have high resistance. So the larger the cycle is, the more resistance we have. The smaller cycle means we have less resistance.
Hi, thank you very much for the talk. But I was wondering, so the goal is to promote axon growth using light in the brain, and from my understanding, egg hydrogels typically have different characteristics in brain tissue, like scattering and transmittance. I was wondering, what are the next steps to making this applicable in like real bio?
So in terms of scattering, it doesn't matter that much. Like we wanted to have a material which does not scatter light that much. Of course, in brain, you have blood vessels, you have other tissues which are going to scatter light, which is slightly tricky. PEG itself, in terms of scattering, we will not have an issue because it's a transparent system. And it's perfect for our application because light can go in. So how we are trying to do is we are actually trying to do it in 3D in after I left the group. So they are still working on it. That first of all, we're trying to establish it in mice. So we are taking the cluster of cells, these neurospheres. So that's why I was showing in later slides the cluster of cells, because of course we are not going to put one cell in the hydrogel. So it's like you take a cluster of cells, put it in PEG hydrogel, and it can actually inject it in the mice, and then it polymerizes during this injection step. And once it's there, then we can shine the light. We have this optical guide actually to see how it looks like. And this work is still ongoing. And we can shine the light just from the top of the skin of the mice. We don't need to remove the skin or anything. And we can really control how this is working. But it's a really long process. Of course, there is blood circulation and so on, which are interfering with our system. But still, this is something that is ongoing, that we are trying to really take it, that we can show it in the mouse as well.
Okay, if there's no more questions, let's thank the speaker.